created by Muhammad Ali, Evan Bristera, and Melissa Fraley. Meiosis and Mitosis. Look at this lizard. At first we see his tail cut off, but somehow he's able to grow it back. How is he able to do such a thing? Well, it is able to do this due to the process of cell division. Mitosis is only one part of what is called the cell cycle. The cell cycle is the journey a cell goes through in order to divide. Because of cell division, we are able to reproduce, divide, grow, and even repair tissues. Now let's have a look at my microscope, baby. It all starts at the smallest unit of life, a cell. What you see here is the entire process of the cell cycle. Cells constantly being copied and divided. Our bodies must produce millions of skin cells in order to replace those we have lost throughout each day. The cell cycle has many phases incorporated into it. The cell must first replicate its DNA so that it can provide an exact copy to each of the two daughter cells that will come from the cell division. After the DNA has been replicated, the chromosomes condense in the nucleus of the cell. The replicated chromosomes are called sister chromatids. The cell cycle is broken up into 4 different phases, taking around 24 hours to complete. Most of the life of cells is spent in interphase. Interphase consists of 3 stages, named G1, S, and G2. G1, or gap 1 phase, is the first growth stage of interphase. In G1 phase, a set of protein cylinders next to the nucleus called centrioles duplicate themselves. Centrioles regulate the movement of many different things inside of the nucleus and cell. In S, or synthesis phase, DNA replication occurs. All the DNA replicates itself, giving the cell 2 copies of every strand of DNA. Next is the second growth phase, G2 or gap 2 phase. In this phase, all the enzymes needed to aid the process of cell division are produced. Now that the cell has duplicated its DNA in the nucleus and duplicated the centrioles, mitosis can begin. Mitosis, or M phase, is split into 4 stages: prophase, metaphase, anaphase, and telophase. During prophase, the first stage of mitosis, we first see the classic chromosome structure, consisting of a sister chromatids and centromere. The centrioles begin moving to opposite sides of the cell, leaving behind protein strands called microtubules, or the mitotic spindle, which leads to a connection from one end of the cell to the other. At the same time, the nuclear envelope breaks down. The microtubules then make contact with each chromosome at a protein structure on either side of the chromosome called the kinetochores. From there starts metaphase. During this stage, the sister chromatids of each chromosome align along the center of the cell at an area known as the metaphase plate. Now the chromosomes are ready to be separated, which initiates the start of the next stage, anaphase. During anaphase, the microtubules attached to the kinetochores shorten and the poles of the cell move farther apart. This forces the sister chromatids of each chromosome to separate and be pulled to opposite poles of the cell. Telophase is the final stage of mitosis. During telophase, the nuclear membrane reforms around each set of chromosomes, and the chromosomes uncoil and return to an uncondensed state of chromatin. A small crease forms between the 2 new cells, known as cleavage, which marks the beginning of the final split. The cell is then compressed by a contractile ring that starts to divide the cell in half. Once the 2 nuclei have formed, mitosis is complete. The only thing left to do is to split the 2 nuclei to form 2 new daughter cells. This is where the final stage of the cell cycle comes in, cytokinesis. In cytokinesis, the contractile ring splits the cell into 2 new daughter cells. By the time the split occurs, the organelles in the cell have been replicated and are divided between the 2 halves of the cell. Once split, the 2 new cells are in the G1 stage of interphase and ready to complete the cell cycle once again. There are 3 different checkpoints that a cell must go through in order to complete the cell cycle. There is one in the G1 phase, another in the G2 phase, and the last in the M phase. The first checkpoint in the G1 phase is late in the phase and is called the DNA damage checkpoint. At this checkpoint, the cell can only move onto S phase if the cell is adequate in size, if the nutrients are sufficient, if social signals are present, and if the DNA is undamaged. The next checkpoint, the DNA replication checkpoint, occurs right before entering M phase. At this checkpoint, the cell can only pass if the chromosomes have replicated successfully, if the DNA is undamaged, and if activated MPF is present. The last checkpoint occurs during mitosis. At this checkpoint, the cell can only pass if the chromosomes have attached to the spindle apparatus. Next, we will discuss the process of meiosis and how gamete cells are able to replicate.
Meiosis is the process in which haploid cells are produced from a diploid cell. For this to occur, chromosomes must be sorted and distributed to create genetically unique cells with half the number of chromosomes as the original cell. For a successful reduction in the number of chromosomes to occur in the haploid daughter cells, 2 rounds of divisions are necessary. These divisions are called meiosis 1 and meiosis 2. One difference in meiosis and mitosis is that mitosis results in 2 diploid daughter cells and meiosis results in 4 haploid cells. Meiosis begins after the cell has successfully completed the G1, S, and G2 stages of interphase. During the S phase of interphase, the DNA is replicated, producing 2 copies of each chromosome called chromatids. The paired centrioles in the cytoplasm duplicate and begin extending microtubules that form the meiotic spindle. Sister chromatids remain attached to the centromere and condense as the cell enters prophase 1 of meiosis. So far, this is very similar to mitosis, but there are 2 events that happen that create genetic diversity. The first event occurs during prophase 1 when homologous pairs of sister chromatids lie side by side in a process called synapsis, forming a tetrad or bivalent. The homologous chromosomes share similar but not necessarily identical genes. Once this structure is formed, the second event, called crossing over, happens. During crossing over, a physical exchange between chromosome segments of non-sister chromatids occurs, increasing genetic diversity. Prophase 1 concludes with the fragmentation of the nuclear envelope as the duplicated centriole pairs move to opposite poles of the cell. As they move, the centrioles expend spindle fibers, forming a mitotic spindle. In prometaphase 1, with the paired centrioles in place, the mitotic spindle is fully formed. The sister chromatids attach the spindle fibers by their kinetochores. Another key difference between mitosis and meiosis occurs because of synapses and crossing over in meiosis. Homologous chromatids remain aligned so that the pair of sister chromatids is attached to only one pole by the kinetochore microtubules. In mitosis, each sister chromatid is attached to a spindle fiber. The spindle fibers originate from opposite poles. Metaphase 1 bivalents randomly aligned along the metaphase plate due to independent assortment. This alignment is random and adds to genetic diversity in anaphase 1. The homologous chromosomes separate and move toward opposite poles. Meiosis 1 ends with telophase 1 when the chromosomes decondense and the nuclear envelope reforms. Cytokinesis separates the cytoplasmic material and 2 daughter cells are separated by a cleavage furrow, since meiosis results in 2 haploid cells. Meiosis 2 begins without the chromosomes going through another round of DNA replication. Centrioles again duplicate and begin moving to opposite poles of each cell. In prophase 2, the sister chromatids condense while the spindle starts to form as the nuclear envelope disappears. In prometaphase, the 2 sister chromatids attach at the spindle by kinetochores, microtubules, with sister chromatids attached to opposite poles. The spindle aligns the sister chromatids along the metaphase plate during metaphase 2. During anaphase 2, sister chromatids separate and individual chromosomes move toward the poles. The entire process ends with telophase 2 as the chromosomes decondense and the nuclear envelope reforms. Cytokinesis occurs and the cleavage furrow separates the 2 daughter cells into 4 haploid daughter cells. The haploid daughter cells will specialize in 2 gametes, either sperm or egg. These fuse in fertilization to form a zygote, which will grow into a child. The child receives half its chromosomes from its mother and half from its father. Because men and women produce millions of gametes, and the selection of gametes in fertilization is random, this contributes to genetic diversity. This explains why a child isn't identical to either parent. And that's